Hi, everyone. Thanks for coming to my session. My name is Feng. I'm a software engineer at Databricks, working on the healthcare and life sciences vertical. And the title of my talk is a little bit vague, but for those of you who are in the genomic space, I'm going to be discussing something called joint genotyping. And that's the idea that when we have genetic variants, there's a certain amount of noise that comes off the sequencing machine. And to get rid of that noise, if you add more data, the more accurate you can identify your variants. So most of you already are probably excited about genomics, hence why you're at this talk. But to give you a bit of an anecdote, genomics has real-world implications today. In 2004, a boy called Nick Volker was born in Wisconsin. And at age 2, he was diagnosed with a chronic intestinal inflammation condition, which doctors called intractable, which is a fancy way of saying we can't figure out what it is, and it keeps happening. As a result of this, doctors went through over 100 surgeries on this boy, including the removal of his colon. When he turned around 5 or 6 years old, doctors decided to go to what they thought was the last possible solution, that he had a genetic mutation. And in 2003, the Human Genome Project was completed, which meant that doctors could compare his genome to what is considered to be the average human genome. And they did what is called whole exome sequencing on Nick Volker. The exome is the part of the genome that directly translates into proteins. So as you can see in this image, when you have genes, those are transcribed into mRNA pieces, which then become protein. If the gene is mutated, then the protein is mutated as well. As a result of this, doctors were able to identify his mutation, which was actually linked to another disease, which had been cured previously via a bone marrow transplant. And a bone marrow transplant is a type of stem cell transplant. So doctors went ahead with this sort of transplant in 2009. And as a result of this, Nick Volker is now a healthy boy. I think he's now around 15 years old, and he has no more immune conditions. So why is Databricks working on genomics? Well, back in the day, being only 10 years ago or so, genomics wasn't a big data problem. Everything that you worked on could fit on your computer or on what is called a HPC, or high-performance cluster. However, as sequencing has become cheaper, the amount of genomic data out there has doubled every 7 months. And that's because in 2008, a new form of sequencing technology came out, and it's called next-generation sequencing, which is a little bit sci-fi sounding, but the idea of it is it's like a parallelized version of the old version of sequencing. And so going from the $3 billion Human Genome Project to the to $100 million after that, now the price of doing a whole genome sequence on a human costs about $1,000. As a result of this, we project that the amount of genomic data out there will explode in the next couple of years. And it will soon be outpacing what is considered to be the normal big sources of data, such as social media sites like YouTube and Twitter. So to give you a bit of an overview of what I'm discussing today, we've now discussed that genomics is a big data problem. However, the amount of data actually enables us to do the real-world applications even better. With the big data, however, we run into the normal pain points that people have when you have a lot of data and legacy tools. One of the tools that I'll be discussing is joint genotyping, which you can guess kind of falls over in the existing architecture. But our approach, which is straightforward and uses a Spark native architecture, doesn't have that problem. So let's start with some real-world applications. We're first going to discuss something of major interest to the pharmaceutical industry, which is getting your drugs from identifying what can be modified with drugs all the way to the market. And drug companies spend millions and billions of dollars on actually getting their drugs to that point. To actually get there, however, you have to go through the FDA. And that can be a long slog of a process. However, if you have genomic evidence in your clinical trials, you can increase that probability significantly. And that's because when you have genomic evidence, you have more of a causal link between what the mutation is and what the disease is causing, and how your drug can mediate that. And what your drug is mediating is what's called a biological target, which is kind of a vague term, but that can go anywhere from DNA to RNA to a protein, anything that can be modified in its behavior with a drug molecule. And to do that, using big data helps. You essentially just do regressions or an association study between the DNA mutations or variants and the trait of interest or the disease that you're looking at. And this paper is one that's done by one of our partners over at the Regeneron Genetics Center. They're one of the largest genome sequencing efforts out there today with over 400,000 whole exomes. And as a result of the amount of data that they have, they're able to find associations that people weren't able to previously. Moving on to another example of big genomic data enabling people to change their lives is the example of precision prevention. And you may have heard in the news recently that Angelina Jolie decided to undergo a double mastectomy as a result of discovering that she had what's called a BRCA1 gene mutation. This mutation is known to have a high risk of causing breast cancer down the line. Knowing this allowed her to try to increase her lifespan by removing the risk of getting this disease. Calculating disease risk can be done via a variety of methods, and in this paper, they discuss what's called a polygenic risk score, which is essentially a weighted sum of whether you have a variant and how high a chance that variant has of causing a disease. And again, how do you solve that? Well, via large-scale regressions. The more data that you have, the better you can make your model when it comes to identifying whether you have a high chance of developing the disease of interest. And finally, there are many other examples for genetics impacting real-world applications, but I'm going to discuss optimized treatment. Specifically, in the realm of pharmacogenetics, doctors have discovered that some drugs have what's called a narrow therapeutic index. Most drugs that you get over the counter don't have this problem, but some, such as warfarin, which is a blood thinner, have a small gap between what is considered to be efficacious and what is actually toxic. So if you take too little, then you'll develop blood clots potentially, or if you take too much, then you'll have side effects, which can land you in the ER as well. And so in this paper that was published recently, you can see that the number of ER admissions that resulted because of people overdosing or underdosing on warfarin has increased over the last couple of years. To fix this problem, doctors have looked, again, at large amounts of data and identified genetic variants which are linked to what is considered to be the right dosage for the drug. So on the bottom here, the words are a little bit small, but those are different genotype variants that you can have. And based on that, doctors can adjust your dosage to figure out what is right for you, rather than prescribe you what is considered to be the normal amount in the first week, and then after that, keep trying to adjust to get you to a stable level. And as you can guess, the more data that you have, the better you can do these regressions to identify what is an ineffective or a toxic dosage based on your genetic variants. So just a bit of an overview of how genomic data has made our lives better today. It has accelerated target discovery, allowing pharmaceutical companies to get their drugs to market quicker. It has allowed patients to improve their lives via precision prevention. And it also has allowed doctors to prescribe drugs at a more accurate level with optimized treatment. However exciting genomic analysis on big data is, it's also a really difficult problem. This is something you encounter in every other field, but the tools may work on the single node machines or on your HPC or your high performance cluster, but they may not work as you start scaling out. In the genomic space, there are a lot of these file formats which are only meant for genetic data. And they're these big flat files. You can have terabytes and terabytes of them. And people expect you to pass them into command line tools, occasionally into R or maybe into Python. Either way, the tools tend to be very inflexible. And so there's this forum for biostatisticians called BioStars. It's kind of where everyone goes to figure out how to get their tools to work. And this person was specifically complaining about how the tools they want to use, specifically the Genome Analysis Toolkit, or GATK, and the other tool they want to use, called VCF tools, or variant call format file tools, their tools expect a different chromosomal notation for the same file format. Specifically, it's essentially just a big TSV in front of which you can put the letters CHR before your chromosome number, or you can remove that. But one expects one form, and the other expects the other form. And so one person helpfully responded with, OK, here's a couple lines of awk to fix your problem. Another person also helpfully responded with, why don't you just learn awk? Every statistician needs to know awk, or else you can't do anything effective. And that's actually kind of a problem. Statisticians who are working on massive amounts of data shouldn't have to just learn how to do awk and set and grep and pipe all these tools together. That's not really their job. So how can we fix this? Well, if you had something in a Spark SQL data frame, for example, you can imagine just doing small modifications on your column, and you wouldn't have to figure out how to, feed yourself for a lifetime with awk. A lot of the tools are also in the single node world, as I've now explained. However, the tools that have to be run don't actually expect you to know the entire state of the file. And what that means is it's really easy to parallelize these tools, even though they exist currently in the command line space and therefore can't be parallelized. This is an example of an algorithm that can be run for variant normalization on these variant call format files or VCF files. And you can see essentially all it really needs is a row in a TSV file. And you can just apply any sort of UDF or user-defined function on your Spark data frame and fix this problem and parallelize rather than just having these 500 terabyte flat file on your HPC. And finally, the existing tools are often stitched together. Most of the tools have a small subsection of functionality that they have, but they all overlap as well. So how do you find the right tool for the job? Well, when you have found them, somehow, you then have to get them to fit together as well. And there is an entire cottage industry of these workflow systems which claim to be able to fit all your tools together. So once you find the right tools, then you have to fit them together. And then you have to call awk, set, and grep a couple of times to get them to work. And there are nearly 250 or so of these on this GitHub webpage alone of workflow systems that you can use. You can kind of see how this becomes a bit of a nightmare. So now to zoom in a little bit more, I'm going to discuss joint genotyping. Not only the existing approach, but also the approach that we've taken over the last couple of months to make this problem amenable to the big data space. So before I go there, though, we're first going to discuss what a DNA variant means. I've discussed genetic variants a little bit now, but to give you a little more context, when we start with a reference genome, that's what we consider to be the average or normal human genome. And at these positions, 0, 1, and 2, there are 3 nucleotides, which are the components of DNA, A, T, and C. There's another nucleotide called G as well. For this sample, or individual or person, you can see that at position 1, the T has become a G. This type of variant is called a single nucleotide polymorphism, or SNP. And this is what a lot of geneticists have been looking at for the last couple of years, although there have been investigations into other types of variants, which I'll discuss later as well. When you have this variant, though, you also have the question of how confident are you in this call? The call being, is this actually a G or is it actually a T? And we're just not super sure about which is which. And that's because the data that comes off the sequencing machine is kind of noisy. And so in variant calling, or identifying what a real variant is, there's an entire pipeline which is recommended by the Genome Analysis Toolkit folks over at the Broad Institute. And I won't go into too much depth on any of them, except for this section on joint genotyping, in which you take your bunch of flat files and then do what's called joint calling. And the idea of joint genotyping is that you're improving these variant calls by taking the massive amount of data that you have and improving your variant calls by essentially taking population statistics. You can think of this in terms of a Bayesian prior from which you can improve your posterior. The more data that you have, the more you can distinguish noise and signal. So look at this row from a variant call format file or VCF file. At this position on chromosome 20, at position 18 million, the reference, or what the normal alleles at that position, is a C. However, it's possible that you can have an alternate allele, or an abnormal nucleotide, called T at this position. And for this sample, which is an anonymized name for someone, essentially, for this sample called HG0096 on the far right, we have strong evidence for the reference allele of C13, which is the first number after the letters AD, which stands for allele depth. The strength for the alternate allele, however, which is after the comma, is an 8, which isn't quite as high as 13. Every human has 2 sets of each chromosome. And as a result of that, you have 2 possible types of variants. You can have CC, 2 reference alleles, CT, one reference and one alternate allele, or two alternate alleles, TT. For this individual, because the allele depth or the evidence for the reference and the alternate allele are essentially similar, we're going to go ahead and call it as a CT, or heterozygous call, one reference, one alternate. However, we're not super sure about this because it's not completely 50-50 either. It's possible in this case that the alternate allele that has been discovered is actually due to noise. And so the quality is a little bit low. It's below 200. And it's possible that a doctor who is trying to identify a variant in an individual in order to treat them might see this quality score and say, you know what, that's not good enough. I'm going to go ahead and throw this out because I think it might just be noise. So how can we improve this variant call? Well, you add in more data. When you have more population statistics, the more confident you can be that this is a real signal. So on the far right, we've added a sample called NA19625, and the allele depth for them, for the alternate allele of a T, is 17, while we are pretty confident that they have no reference allele, hence the 0 before the comma. And as a result of that, we are very confident that the alternate allele exists, and you can see the quality score go from around 200 to nearly 700. And as a result, we may not throw away this row when we're doing actual variant calling to identify whether this person has some sort of disease that may be linked to this variant. So we have so far discussed one type of variant, as in the previous example, where a C became a T, of a SNP, or a single nucleotide polymorphism. There are many other than the ones I'll be discussing here, but we're first going to discuss insertions, where at the positions between 1 and 2, sample 2 has had 3 A nucleotides inserted. You can also have a deletion, where this individual, sample 3, has had the T nucleotide removed, and so they go straight from A to C. And these two types of mutations together are called indels, or insertion deletion. Why am I discussing this? Well, I want to show you some accuracy metrics of how joint genotyping can really impact how your variant calls are made. Specifically on the two types of variants we've discussed so far, insertion deletions and SNPs, or single nucleotide polymorphisms. There are two accuracy metrics which I'm discussing: recall, which is true positives over the number of true positives plus false negatives, which is essentially how much of the real signal did you recover and think was true signal. We're also discussing precision, which is true positives over true positives plus false positives, which is how many of what you thought was real signal is actually real. And so on this accuracy chart, you'll see some pretty high metrics. Generally speaking, the recall and precision for both types of variants is above 96%. However, we can improve that. And as I've now mentioned multiple times, you can do that by adding more samples. When you add more samples, you can increase the quality of your variant calls and your accuracy as a result. So when you add 2 more samples to this lone sample HG002 as part of a trio, you can get the recall for insertion deletions all the way up to 98%, which is a 50% improvement in accuracy. So you can imagine that everyone wants to do joint genotyping. You can improve your variant calls and anything downstream of that, including your association studies, et cetera. But what if we added even more than just having 3 samples? What if you had 1,000 samples? What if you had hundreds and thousands? Well, the existing architecture just doesn't really allow for that. So we're going to allude to what is considered to be the gold standard, called the Genome Analysis Toolkit architecture for joint genotyping. First, we start with a bunch of flat files called GVCFs, or Genomic Variant Call Format files. These are then piped into a command line tool called GVCFs, which then pipes out another GVCF with all of the samples included in it. This is then piped into another command line tool called Genotype GVCFs, which spits out what's called a Project Variant Call Format file, or PVCF. And as you can imagine, flat files can become a bit of a nightmare. If they don't fit on your high-performance cluster or your laptop, then everything just kind of explodes. And also, the command line tools themselves have some inherent problems. Combined GVCFs has an exponential runtime. And what that means is the number of files that you add in will make combined GVCFs take longer and longer, to the point that as you add in tens, hundreds, thousands, hundreds of thousands of samples, you just won't be able to finish. And also, it has what's called the N+1 problem, or the incremental problem. People who are dealing with genotype data don't usually just get 1 million samples completely genotyped, and that's the end of the day. They get batches of data. And if you add a single sample, you have to rerun this entire pipeline from scratch because you can't just add it to the GVCFs. It's a pretty complex file format from which you can't just slap it on the end as an extra column. Also, this tool, as a command line tool, runs in a single node environment. And there are ways to parallelize that, which I'll discuss in a little bit. But I believe that with Spark, you can run this in a much more efficient way. And again, another command line tool runs in single node downstream called genotype GVCFs as well. And this causes an inherent limitation when you're dealing with big data. So as I've mentioned, there are attempts to parallelize this existing architecture. And this is in a language called the Workflow Development Language, or WDL. And this tool that they've used is called scatter-gather. And this may look familiar to you, and I'll mention why later. But the idea is you start from a flat file, you split it into a bunch of smaller flat files, those get piped into a command line tool, and then those get stuck together using some sort of bash tool to create a final file in the gather step. However, when you're trying to do scatter-gather, you run into a couple of different problems. First of all, there's inherent data skew that is introduced when you do what is considered to be, I would guess, the most common approach, which is splitting by chromosome. Humans have 24 chromosomes, but they're actually of different sizes. So you don't even get a 24x speed up when you do the splitting. You get way less than that. And that's because something like chromosome 22 is nearly 5x smaller than something like chromosome 1. You can also split by interval, essentially chopping up the chromosome into smaller pieces, but this requires you to create a hand-curated list according to your reference genome to pass as a command line argument. Either way, you're still in the single node space. In addition, you have to learn an entirely new language called the Workflow Development Language, which is then piped into something called a Cromwell server, which only runs on a limited number of cloud platforms as well. Well, what if you were able to fix all these problems without learning all these new languages? Well, you can use something like, Spark, and that's why we're all here. Scatter-gather looks a lot like MapReduce, but we don't have to deal with all the flat files in between. And this is the core of what I consider to be probably the simpler architecture that we've built out here at Databricks. And let's break down the genome analysis toolkit architecture into 2 steps. The first step is essentially just variant ingest. And as we've mentioned so far, there are a couple of different problems with the existing architecture. You have a lot of flat files in the GVCFs, which need to fit on your machine. And combined GVCFs has an exponential runtime. It doesn't work well for the incremental case, and it runs on a single node. So in the Databricks architecture, you can get your data of the GVCFs either from your flat files or from Parquet or Delta or something like that. Either way, when you load it in, that takes a linear runtime. And that's because we're not doing any of the combining logic here. We're doing it downstream because that's required specifically for the GATK architecture. And this is really straightforward. Reading from Delta, you would essentially do the same thing, but replacing com.databricks.vcf with Delta. But because the VCFs are essentially just big TSVs, we can load them into a data frame, split the files up, read them in parallel, and you get this big data frame with all of your flat files combined. And now that we have this linear runtime on data that is partitioned across multiple machines, we can then do parallelism in a way that fits well for our downstream algorithm without going through all the complications of scatter-gather. So in this step, in this one-liner essentially, we call a user-defined function called bin, which just attaches bin IDs to each section of the VCF so that overlapping sections are in the same partition so that the downstream engine for the GATK is able to operate them properly. And for those of you with a genomics background, this is because of something called spanning deletions, for which you have to be stateful. And in the final step, for variant ingest, we can solve the problem of n+1 and the exponential runtimes earlier with the incremental adding to delta lakes, which is a linear runtime as well. And this is something that has been approached in the space by other approaches, such as Genomics DB, if you're familiar with what the GATK has been up to recently. But that's a pretty complex architecture created by the Intel and GATK teams. And it's something that we can solve essentially for free by just using Delta. And this is also essentially just a one-liner. We just call something like write with the append mode to Delta. We can partition by according to what the downstream engine needs, and then just save it in our Databricks mount or in something like S3 or Azure Buckets. And so that's essentially it for stage 1 of ingest. This essentially looks like 3 lines. And in the real world, it's not that much longer. It's only like 100 or so lines. I do comment very aggressively, so this is kind of an overstatement. And in the second step of the GATK architecture, we essentially just do the actual re-genotyping, the exciting step. We're not just ingesting data anymore. And in the previous architecture, we pointed out a couple of different problems. We start from a flat file, which doesn't play nice with the n+1 mode, or when you have more genotype batches coming in. We also have to run on a single node command line tool, genotype gVCFs. And again, we output to a single flat file of a pVCF. Well, Delta Lake has already solved all of our incremental problems. And to read in from Delta, all you need to do is just call load, and you get this big data frame on which you can do all of your processing. The second step is we just do the fine-grained parallelism needed to call into any joint genotyping algorithm. Some can probably process row by row, but here we're currently calling into the GATK's tool, genotype gVCFs, which is well regarded as the gold standard for joint genotyping. And this is a bit of an oversimplification, but the idea is you could just call something like map partitions on data that has been partitioned in such a way such that your engine can process the data properly, and then just create a joint genotyper which can have a complex state that you don't have to worry about too much, and then just genotype that row. And that will essentially be output as a regenotype row or a pVCF row. And finally, when you have your big data frame of these project variant call format rows, you can then output these to a flat file of a pVCF, if that's what you so desire, for downstream querying. Or you can put it in Delta or Parquet or some other sort of database which is easier to query quickly. And not only do we allow you to read in from VCFs, we also allow you to write. And so, again, just another one-liner. And so with all of this logic, we just break it down into 3 simple parts. And again, with all of my aggressive commenting, we're still below 300 lines of logic. Joint genotyping is something that I've been really excited about, something I've been working on for the last couple of months. And I'm excited to say that I've completed running the 1,000 genome project on it in less than 24 hours. And that's because it runs quickly across scale with more and more samples. Here on the bottom, in a log-log scale, I have the number of samples and the runtime in minutes. On this scale, it's a linear runtime. You might notice a bit of an elbow, and that's just because we ran into what's called a spot termination. And that's because I was being cheap. Instead of using on-demand instances on AWS, I used spot instances. And I got outbid at the last minute during my variant ingest, so I had to restart that, just restarted the ingest step, which made the runtime run up a little bit. But it just means that, we don't have to restart from scratch for everything. It was just that one worker node, and so we're tolerant to any sort of faults that happen in the system. Also, we scale well in terms of cluster hours. So when we scale across the number of workers and increase them, the runtime also decreases linearly. Here, I was using a really small data set. It was just 10 samples from the 1,000 Genomes project. So by the end, with my fairly large bins, I was kind of running out of CPUs that I was actually using. But this does scale generally linear on this log-log scale for runtime. And finally, it is GATK-concordant with what is considered to be the gold standard. If you ran combine and then regenotype on the command line, you get essentially the same results. We've mentioned earlier recall and precision as accuracy metrics. And in this chart with the rows, we get more and more specific in terms of what we're looking for from site to genotype to calls. Either way, the numbers are essentially 100%. And if you're going to nitpick at the last 0.001%, well, that's because there are parts of the genome that are really hard to genotype with our current algorithms. Our variant callers just aren't able to account for these sites. And if you're a geneticist, you might be familiar with the term highly multi-allelic, and these are the sites that the GATK also has problems with. So I believe the work here is pretty much close to done, and there's a little bit more that I think we could do. I've mentioned in between that there's a joint genotyping algorithm from the GATK. And this is something that we can improve upon, I believe, in the space of genetics. And that's because pharmaceutical companies and everyone else doing analysis are interested in what's called a rare variant. A rare variant is something in your genome which only happens in a small proportion of the population. However, there's a lot of people, and so even if the variant is fairly rare, it will occur multiple times across the number of people, and these can have a massive effect size. And this is essentially just one step in the process. We can just swap this out, essentially, and change the parallelism to work well for that algorithm. So if you wanted to account for rare variants, you just switch out the map partition step. And finally, I believe that there are approaches to be made to make this algorithm more amenable to incremental updates. Right now, I'm actually calling partition by after reading in from Delta, which requires a bit of a shuffle. But you can do the partitioning as you're writing into Delta, as I described earlier, and then just use Delta optimize to deal with small files a little bit better to kind of smash them together. And finally, I'm just going to give a bit of an overview of how Genomics fits into the Unified Analytics Platform at Databricks. So on the Genomics side, we have a bunch of flat files, which people usually want to do processing on, such as BAM, CRAM, and SAM, as well as the variant call format file, VCF. These can be ingested using our Spark data sources into data frames and used in sequencing pipelines such as DNA, RNA, and tumor normal sequencing, or cancer sequencing. These can then be put in Delta and used in tertiary analytics tools like joint genotyping, as I've discussed, as well as association studies for finding associations between variants of interest and a disease. And finally, these can be put in dashboards, or you can do your cool machine learning technique on this data as well off of Delta. And on the DNA sequencing side, we have a blog post about this where we were able to run on a massive data set of 300x coverage. This may not mean a ton to you, but the idea is that you wouldn't be able to run this anywhere else. And because we're able to parallelize by calling into the GATK in a Spark native way, we don't have the inherent limitations existing anywhere else. And one of my other teammates has created a really cool blog post about a dashboard he made for variant data. As you get more data and you put it in your Delta Lake, you can then analyze the variant data that you have in real time. And this fits into a larger healthcare and life sciences project at Databricks. It's not just genomics data. We also do medical imaging and et cetera in the pharmaceutical, providers, payers, and government space. And that's basically it. If you have any questions, let me know. There's an entire blog series on genomics on our website, and you can sign up for a private preview as well. And I've been told by my moderator that you can ask questions and also to rate and review the session on the app. So thank you so much. If you have questions, you can go to the middle, or you can leave.
Thank you for a very nice talk. So one of the problems with taking the VCF format and doing large-scale computation on it is it keeps a whole bunch of extra data around that you don't necessarily need. So I wonder if you've looked at more sparse formats like the Sparse project VCF, which can scale that type of computation much more linearly.
Yeah, so just to repeat the question, the VCF file format is pretty well known as being a little bit overly verbose, and it can be like really memory hungry. So how can you solve that using something like sparse VCFs, which are a little more amenable? Well, you can deal with something like Parquet if you want to compress your data a little bit more. You can also do higher level functions in Spark SQL to get rid of information that you don't need. Or you can put it into something like MLlib and use sparse vectors and matrices on top of that. So in the Spark architectures, you don't actually have to go that far. I think that there are definitely methods that exist outside of the Spark native space, which can allow you to maybe get small improvements on top of that. But it's not something that we're looking into right now.
OK, yeah, I just wanted to point you at It's an actual standard sparse project, VCF, which...
Yeah, I actually haven't looked into the sparse VCF spec too much.
Okay.
It's not something that has come up at our customers recently.
Okay, great, thanks.
Mm-hmm.
Thanks for the talk. Really cool about the N+1 stuff. Just wanted to defend the Brodies. It's the workflow definition language, not development.
Oh,
and they
sorry.
are adding support for Cromwell for AWS in addition to GCP. Question, do you plan to get up in the graph genome game?
I actually don't know if I have a good answer for you on that. It's not something I'm working on actively right now. I definitely think the stuff that Broad is doing when it comes to GCP and AWS is really cool. I think it's just something that we felt that was really easy to parallelize on top of the way that Spark works on Databricks today, without recreating an entirely new language on top of that. But I think the work that is happening there is fascinating, and I can't say that we're looking into that ourselves right now.
Okay. And then I just want to clarify, like claiming solving the N+1, that's like huge. So you're saying that you can do your genotype calls without recalling your entire cohort? Or could I just get like a 2-sentence on that?
So, I mean, we are recalling the cohort in the sense of you need to obviously do joint genotyping on the data set. It's removing the exponential step from combined GVCFs, which I know the Genomics DB folks at the Broad have been doing as well. It's just essentially pushing it downstream so that you don't have to do the extra combination up front.
Thank you.
Mm-hmm.
Hi, very good talk. So I have 2 questions. The first is about the joint genotyping that you do. How do you integrate with the existing tools, or did you have to re-implement a separate implementation to actually integrate it with Spark? Because I noticed you did RDD or data frame map partitions.
Yeah, so to repeat that, the question is how much did we basically have to do on our side to get joint genotyping to work? Well, we're actually piping directly into the GATK's engine of joint genotyping with minor modifications so that we don't write out to a VCF file immediately. Essentially, we're just calling map partitions, and we're creating a new engine. We're combining the rows that are relevant for that, putting it into the engine with the .genotype call, and then taking the output and putting that into another data frame with flat map. So it's actually not that much logic on our side that does the joint genotyping itself. It's just calling other tools. And so that makes it easier to switch out for something else.
That's very cool. Just to follow up on that, is that similar to sort of the RDD pipe functionality?
I actually can't say I have an answer to that. Unfortunately, I didn't really work with RDDs that much. I kind of came on into the Spark SQL data frame scene. So
sure.
I'm sorry if I can't give you a better answer.
Yeah, no worries. So the second question is about enriching your data sets with annotations. How much of that have you done, and what are the challenges there?
By annotations, do you mean variant annotation, or?
Yeah, variant annotations are just other types of annotations you want to add to your data set, phenotypes and things like that. Yeah,
so that's something that you can put in your data frame if you want to do it manually. We can also call into something like SNPF or the other common variant annotation tools using the pipe API, which exists in Atom, which is an open source genomics tool as well. So we can do annotations in the VCF file format, or if you just want to add columns to your data frame like you would and not really live in the VCF world anymore, it's also an option.
Awesome. Thank you.
Yeah. Okay, I think that's the end of my time. So if you have any questions, feel free to ask me later. Otherwise, thank you so much for coming.